Okay, hello to all of you. So we have reached to the point where we are going to discuss the last lab of our mini project. So basically in the first two labs, we have, in the first lab, we have isolated protein by using ammonium sulfate precipitation, and we have got the mixture of proteins. And in the second lab, what we have done, we have passed the mixture of the proteins over the column, and we have tried to purify the protein. Our desired protein is Rubisco by the technique called ion exchange chromatography. So basically, we pass the mixture of proteins over the column, and the column is positively charged, and our protein of interest, which is Rubisco, is negatively charged. And that protein goes and sticks on the column, and then we wash the column, and then we do the elution. We have used low salt buffer, medium salt buffer, and high salt buffer. And these low salt, medium salt, and high salt buffers basically contains sodium chloride in it. And how does the elution works? How the proteins are eluted from the column. So basically, chloride ions, they just go and take the place of the protein. So the protein is replaced by the chloride ions, and the protein basically gets off the column, and we collect the protein in the fraction, and you were taking the OD of the protein in the spectrophotometer, right? So basically, this technique is called ion exchange chromatography because the chloride ions, they are getting exchanged for the protein. So now the column is positively charged. The chloride ions, they're going to go and bind to the column, and proteins are going to be separated. And that's what we did in the last step. So now what we are going to do is we are going to run the protein gel, or we are going to use the technique called protein gel electrophoresis. So basically, we are going to run the gel, and you must have run the gels earlier in the lower division classes. You must have done the DNA gels, and the DNA gels are basically horizontal gels. This is a vertical gel. So this technique is called protein gel electrophoresis. So we will be basically, now we have collected the fractions, and for you guys, all the fractions are there in the fridge. We have, for every group, we have filtrate, we have S1, we have S2, we have P1 before purification, we have P2 before purification, and then we do have P1 and P2s that are eluted using the medium salt buffer and the high salt buffer. So we have collected all these fractions, and now what we are going to do, we are going to run them on the gel, and we are going to check the protein in them. And our desired protein is Rubisco. And Rubisco, basically, when it runs on the gel, it basically shows two polypeptide chains. One is 55 kilodaltons, and another is 14 kilodaltons. So now the very first thing all of you will be doing will be, you will be taking out your samples, and then you will be deciding. Basically, everybody is going to run their filtrate. Everybody will be running P1, everybody will be running S1, S2, P1, P2 before purification, and we will see in the lab. We will go and check your data, which fractions you have eluted from the low salt, medium salt, and the high salt buffer. We will see the fractions which are showing good values. We will choose one or two of them, and we will be running them on the gel. So let's first of all talk about how this technique works, and what does this technique tells us, basically what information we will be getting by running this gel. So now we want to see if we have actually purified Rubisco or not. Okay, first of all, we want to see if we have isolated Rubisco and if we have purified. So that is why we have been keeping all the fractions at different stages. So how does this technique work? So basically, this protein electrophoresis is called STS page. Okay, what is STS? It is sodium dodecyl sulfate. Sodium dodecyl sulfate is a detergent, and you will see that it gives negative charge to the proteins. It masks the surface of the protein and gives the protein negative charge. So you know that there are different kinds of amino acids. The positively charged amino acids, negatively charged amino acids, polar, non-polar. What we want to do in this technique is we want all the proteins to become negatively charged. so that when we run them on the gel, because you know that in this gel we are going to apply the electricity and there will be two electrodes. So we are going to load our samples, and we will like our proteins to run towards the positive electrode. Because now the proteins are masked by STS, and this STS gives negative charge to the proteins, these negatively charged proteins will be running towards or moving towards the positively charged electrode. So this is the purpose of using STS, because it gives negative charge. And when the proteins, all the proteins get negative charge, then in the gel they are going to run on the basis of their size. Any protein which is bigger in size will run slow, and that protein will be near the well. And any protein, I will tell you, like, we are going to denature the proteins also. So in the gel it will not be a protein anymore, they will be polypeptide chains. If you go back and review the structure of protein, you know that proteins have a shape. They are folded, and the protein takes the three-dimensional shape. But in this technique, we will be denaturing the proteins. Okay, STS itself helps in denaturation, and we will also be using heat that will also help in denaturation. We want to denature the protein, we want the protein to get converted into polypeptides, and these polypeptide chains will be getting negative charge by STS, and we will be basically, when we will be running these denatured polypeptides in the gel, they will be running towards their positive electrode, and also they will be running on the basis of their size. The polypeptides bigger in size, they will travel less and they will stay closer to the well. The polypeptide chains, which are very small in size or small in size, they will be traveling more distance in the gel, and they will be running away from the well. So this is the main criteria of STS page. And there is one more step which we'll be doing. Before we actually run our samples on the gel, we are going to mix our samples with a sample buffer, and I'm going to discuss what is there in that sample buffer, and we are going to heat them. Okay, so let's see. Basically, because we are not quantitating our protein, so we will be, for every sample, we will be taking around 50 microliters of our sample, and we are going to mix that with 10 ml of the sample buffer. Now, what does the sample buffer contains? So it will be ready-made given to you. You have to mix 50 microliters of each sample. Each sample means your filtrate, your P1, P2, S1, S2, all the different things. 50 microliters of that, and you will be adding them to 10 microliter sample buffer. Now the sample buffer contains a dye called bromophenol blue, glycerol, dithiotritol, SDS, which is a detergent. A reducing agent, and reducing agents, as you remember, like in the proteins, there are disulfide linkages. There are bonds, covalent bonds between sulfurs. Dithiotritol is a reducing agent, and it can break the disulfide bonds. Okay, this is an important question for your exam. Then glycerol we add so that our sample becomes a little heavy. Now STS is a detergent. Now what does STS do? STS detergent denatures the proteins, because we want proteins to get converted into the polypeptide chains and adjust their overall charge to become negative. So STS is serving two things. It is denaturing proteins. It is also giving negative charge to the proteins. This allows for the proteins to be running in the gel on the basis of their size. And also we will be heating these samples. So as I said, you will take 50 microliter of each sample, and you will be mixing it with 10 ml of the sample buffer, and then you are going to heat your samples on the heating block. The temperature of the heating block will be maintained at 95. You are just going to heat your samples for around 7 minutes, and more I will tell you in the lab. So after 7 minutes, our samples will be ready, and then we are going to load our samples on the gel. So I will show you in the lab how to, how to fix your gels in the apparatus, and we will be like fixing our gels in the apparatus, and then we will be given a running buffer, and we are going to put that running buffer in the apparatus, and we will load our samples and we will run our gel. And also, when very very important thing is, when you run your samples, you also need to run 15 microliters of a marker. Okay, so it's called ladder, we will be giving you ladder also. So along with your samples, all the samples you will be running on the gel, you will also be loading, along with your samples, you will be also loading a molecular weight marker. And the purpose of using this molecular weight marker is because it is like a reference. Okay, so I will tell you, when you load this marker on the gel, eventually when the gel will be run, we will be staining it and then we'll be de-staining it. You will be able to see some bands in that lane, and we already know, because this is a commercially available marker which we buy from a company. The company tells us the first band size is 100 kilodaltons. I'm just saying that. Okay, I will tell you exactly tomorrow what is the size of each band. Then the size of second band is this much. Then the size of third band is this much. So this is a kind of reference in our gel which can actually tell us if we have Rubisco or not, because we know that Rubisco has two polypeptide chains. One is 55 kilodaltons, another is 14 kilodaltons. So we, if we have actually isolated or not, or if we have actually purified it or not, how to know that? By comparing the bands in the marker. If we have a band which is coming near to 55 or another band coming near to 14, that is a sign, yes, we have isolated or purified Rubisco. So the purpose of using marker is, it's a reference. It tells us, it gives us an idea about the size of the bands in the gel. So you will be loading around 15 microliters of the ladder, and the other samples will be, you will be taking 50 of them, mixing with 10 ml of sample buffer. You will be eventually having around 60, but the maximum capacity of these gels, the well in each gel is 50 microliters, so you will be able to load 50 microliters. So after that, we are going to connect our unit, electrophoresis unit, with the power supply, and we are going to run our gel at 100-degree volt for about 50 minutes. And after your gel is run, we are going to take your gel out from the so the gel will be inside these plastic plates. We'll be taking our gel out, and then we are going to stain it with Comessi blue, and then we are going to de-stain it. And so after you de-stain, you will be taking its picture. So more information I will telling you in the lab, sometimes it takes a little more time to de-stain. So maybe you guys have to come to the lab and check your gels, and eventually when your gels are fully de-stained and you see nice bands, you're supposed to take the picture of the gel, and this picture of the gel will become part of your lab report. And when our gel will be running, we will be discussing how to write a lab report. So from these three labs, you have to write a lab report, and lab report will be written as a group. Okay. So the discussion topic for this week will be discuss the application of the technique STS page. Also discuss why we have denatured proteins before loading them into the gel, and why we are using a protein marker with other markers in the gel. So these are important questions I've written here, because these kind of questions can be there on your exams. So that's all for the instructions, and more instructions I will give you in the lab.